The role of astroglial water transport in perivascular CSF interstitial fluid exchange. Perivascular spaces surround both arteries and veins at the cortical surface, creating a pathway for the movement of CSF through the wider subarachnoid space and into the brain parenchyma. Dynamic imaging approaches demonstrate that CSF tracers move rapidly through these spaces, facilitating the rapid exchange of CSF and interstitial fluid. The glial limitans faces the subpial space at the brain's surface and surrounds the perivascular spaces of penetrating cerebral arteries and veins. Interparenchymal vessels, in turn, are ensheathed by astrocytes that completely cover the cerebral microcirculation with overlapping end-foot processes. At each of these surfaces, the gateways of the brain interstitium, astrocytes express high levels of the water channel aquaporin 4. This can be seen clearly in this photomicrograph showing aquaporin 4 expressing astrocytic end feet in purple surrounding the entire interparenchymal vasculature. Immunoelectron microscopy shows that the vast majority of this aquaporin 4 is localized to the end-foot membrane facing the endothelial cells of the blood-brain barrier. Based upon this pattern of localization, it appears likely that aquaporin 4, which bounds perivascular spaces along which CSF enters and interstitial fluid exits the brain parenchyma, may serve to organize fluid movement within brain tissue along the axis of the cerebral vasculature, supporting the rapid distribution of solutes within the CSF and clearance of solutes in the interstitial compartment through a process termed glymphatic exchange. This hypothesis has been tested experimentally in mice that have had the gene encoding aquaporin 4 knocked out. The rate of perivascular influx of CSF into and through the brain interstitium can be measured by infusing fluorescent CSF tracer into the CSF of the cisterna magna, and then imaging the movement of the tracer into the brain at defined time periods after tracer infusion. In animals that express normal amounts of aquaporin 4, CSF tracer moves rapidly into and through the brain interstitium. However, in mice lacking the gene encoding aquaporin 4, the movement of CSF tracer into the brain parenchyma is dramatically reduced. The effect of aquaporin 4 gene deletion upon the clearance of interstitial solutes, including amyloid beta, has also been evaluated. When radiolabeled tracers, such as mannitol or recombinant amyloid beta, are infused into the caudate nucleus, they are cleared from the normal brain at certain defined rates. However, when these traces are infused into the brains of aquaporin 4 knockout mice, the clearance of both mannitol and amyloid beta are markedly slowed. These findings suggest that perivascular aquaporin 4 facilitates the movement of CSF into the brain and the clearance of interstitial solutes, including amyloid beta, along perivascular spaces. This glymphatic system, this brain-wide network of perivascular spaces, coupled to astroglial water transport, plays an important role in maintaining the proper composition of the brain interstitial space.